hydration or hydrolysis? Good. And you do that because you split out what?
You split out water.
Very good. Y'all did study. When you put monomers together or any polysaccharides, if you're joining a monosaccharide to something, what kind of bond is it? A covalent bond, right? And what do we call it specifically? It is covalent, the glycosidic. Anytime you see glycosidic, it's going to be between carbohydrates. And we talked about how the starch was the storage for plants, and we can get energy out of that, and glycogen is how we store polysaccharides. And I also told you that we store it in the liver and the muscle cells, and your storage of carbohydrates only lasts for about 24 hours. Then we moved on to structural polysaccharides. The first one we talked about was cellulose. We said all those things about cellulose, that it is a polymer of glucose. Can we break it down though? If it's got glucose, 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 we like glucose, and we say, yes, I got glucose, because I've just now taken in some cellulose. Doesn't matter, we can't break down those bonds, remember? Animals cannot break down the bond in cellulose, so we do not get glucose from that. Does that mean we don't need glucose in our diet?
No.
We do need it, right, and why do we need it? We can't get energy from it, but what's the purpose of it?
Doesn't it like help build?
It keeps our
It's fiber, and you know what, it's kind of like, you just send this bulk through your intestinal system, and it's gonna kind of help clean it out. It does make you feel full and things like that because it's substance, you know, it's not gonna get broken down very much. So we do need it, but when you look on your package contents and it says insoluble fiber, that's really, they just don't put cellulose because, you know, I guess people like the word fiber better. Okay, we talked about all that. We said though, if you are a fungus, you will have structural polysaccharides in a different form. Fungus do not have cellulose, they have chitin, C-H-I-T-I-N. It's found in the exoskeleton of spiders and insects. This kind of looks like an out-of-body experience for them, but that's how they molt. Also, chitin, number 2, is in the cell wall of fungus. And, which I think is rather interesting, chitin is used sometimes to make strong and flexible surgical thread. Who would have ever thought to use chitin for surgical thread? I got confused one time when I was making a multiple choice quiz or test, I don't remember which, and I put one of the choices was it in, you know, because I know it has to do with insects, and then I put insects use chitin to make surgical thread. And so somebody came up and said, do insects really make the, you know, use that to make, and I said, well, you know, if you put that, you could argue I would have to count that right. That's a mistake on the paper, but I don't really know. How can we say insects don't? It's one of those things I can't prove it, that it's not true, because I have not observed every insect. So I don't really know what insects do in their spare time. I don't think anybody has observed it, but I still don't think you can say they don't. Some people do. I think it's people, but anyway, I think that's corrected. You didn't even know it's made surgical thread. Okay, so now then we'll go to the next class, which are lipids. Now remember, lipids are the ones that we do not consider polymers or macromolecules. The important things to know about lipids is that they do not mix with water. You know why they do not mix with water? It's because they are, what, is water polar or non-polar?
Non-polar, or wait,
They're
polar.
polar.
Polar, so they are,
Non-polar.
right. If it's non-polar, it's gonna have a hard time mixing with water. And it says here they are hydrophobic because they consist mostly of hydrocarbons, which form non-polar covalent bonds. And if you know, I mean, you all already know if you walked in here that oils and water are not soluble. You know that. So if you want to stop and think, why is it not? Well, you probably already remember that water is a polar covalent bond, so it's just the opposite. And so if you can remember water's polar, then you know that it's non-polar if you know it doesn't mix. The most biologically important are the fats, phospholipids, and steroids. Fats are constructed from 2 different types of molecules, glycerol and fatty acids. Fats are constructed from glycerol and fatty acids. Glycerol is actually a 3-carbon compound. I'm not going to expect you to remember that about glycerol. but I was just for your information. If you have 3 fatty acids attached to the glycerol, then that is triglyceride, which you've heard about. This would be triglyceride if you check for in your blood. You know how we have glycosidic linkages when we're talking about saccharides, joined polysaccharides? We don't have a glycosidic linkage. It is a covalent bond. And look, we are splitting out water. Same thing, dehydration. But the bond that forms in a fat is called an ester bond or an ester linkage. I think at the end of your chapter, it asks you to fix a little chart and say each of the different type subunits, say what type of linkage it is. So for you, that we would have, for the carbohydrates, they have glycosidic. For the fats, we have ester linkage. Okay, there are 2 classifications of fatty acids: saturated and unsaturated. Saturated are fats that are solid at room temperature. You can remember that, saturated as solid. An example of this is butter. It is fully saturated with hydrogens, thus there are no double bonds, they're all only S single bonds. Saturated has all single bonds in the chain. I'll show you what I'm talking about. In the chain, only single bonds. They are solid at room temperature, and they have the most number of hydrogens that it can hold. And because it's got so many hydrogens, that's why we don't have a double bond there. Unsaturated have a double bond. Unsaturated fats at room temperature are liquid. An example of that is vegetable oil. They cannot pack together close enough because of the kinks, K-I-N-K-S, in the little chain. Let me show you. Okay, here is a saturated fat molecule. You see, there are double bonds here, but I'm talking about in this little chain. Each time you have like this little peak, that means carbon, carbon, carbon. Now they did not put in the hydrogens, but because there is no double bond there, I know that on this, attached to this carbon, there are 2 hydrogens. Attached to this carbon, there are 2 hydrogens. Let me show you what I'm talking about. So we have this chain like that. Each one of these, that stands for a carbon. So this carbon is attached to this carbon, and it's also attached to this carbon, but that's only 2 bonds. How many bonds does carbon have to have? 4. So I know that there's a hydrogen here, and this, each carbon that we see has 2 hydrogens attached to it. Now we've got 1, this one has 1, 2, 3, 4. Do you see? They just didn't put the hydrogens in, but each one has 2 hydrogens. This is a saturated. So that each carbon has the 4 bonds.
So the carbons are each time it goes up.
Yes,
And
or
down.
down.
Up
Each,
and down.
this is a carbon, that's a carbon, yeah, that's a carbon. Now, carbon's gonna have 4, so each time that we have a carbon, 2 of them, it's like attached to each carbon on one side, and then it's also gonna have 2 carbons, 2 hydrogens that it's attached to. Now, if you have unsaturated, let's say we have this, wonder if it doesn't have this. So this carbon, if we have this though, let's see, we have 1, 2, 3, 4, if we have a double bond. I've taken out some hydrogens. And when I take out the hydrogens, the carbon's not going to be happy because it's not going to have 4 covalent bonds. So what it does is it forms a double bond with another carbon. Now, because of this double bond, it can be in different places in the chain. But like you see this one here, okay? That gives a bend to the molecule, and because of that, it can't pack in close enough to stay solid at room temperature. That's why they're liquid. And we call it unsaturated because it is not, if something is saturated, it's got all it can hold, right? This is not saturated. What's it not saturated with?
With the hydrogen.
Exactly. It's not saturated with your hydrogens, you're right. Now, if I have an unsaturated like this, could I take it if I have an unsaturated fat? Could I make it a saturated fat?
You could add some of that hydrogen to it, right?
Exactly. She said, what about if we add some hydrogens? Well, if we add hydrogens, then what's going to happen that double bond is gonna break. And we're gonna go back. Now we have a saturated fat. You know what we say? We say that that has been hydrogenated. Because if you look at the word hydrogenated, what's the first part of that word? Hydrogen. If you want to say hydrogenated to yourself, that's okay. That means you've added hydrogen to it. Now that you've added hydrogens to it, it's saturated. Hydrogenated vegetable oil, it's unsaturated fats that have been synthetically made saturated by adding hydrogens. Example of these are peanut butter and margarine. They are hydrogenated to prevent the lipids from separating out in oil form. Some peanut butter is not hydrogenated. Have you seen peanut butter that the oil separates out on the top? Yeah. And you have to get it and you have to stir it and mix it up together? That's because they have not hydrogenated that peanut butter. Okay? Let's see, most animal fats are saturated. Plant fats and fish fats are usually unsaturated. If you want a healthy diet, which one do you think would include? Saturated fats or unsaturated?
Unsaturated.
Unsaturated. Animal fats are not healthy fats, and they can lead to, I put on my sheet atherosclerosis, but if you just want to put cardiovascular disease, it's just atherosclerosis is a cardiovascular disease due to plaque deposits because that excess lipid gets deposited in your arteries, and of course, it's if that plaque breaks loose, you can have problems there. You can have a heart attack, or it can go to your brain and have a stroke. Okay, you don't have to know the difference in the trans or the cis. But, you know, fat, sometimes, I don't know, I just don't like that word fat. Do you? Just saying it is kind of disgusting. But it's really, we need fat. There are purposes in our body for fat. And the first major function would be for energy storage. If I were to say, I want the most energy I can have because I've got something really hard to do, and I can have 100 grams of fat or 100 grams of carbohydrates, what do you think I should take if I want the most energy possible? I should take fats. Fats have 2 to 3 times as much energy per gram as carbohydrate. Now, carbohydrate will get fed through faster. It's going to give you a faster surge of energy, but it's not going to last as long. So, you know, if you want to get a good start and keep going, then you'll probably want carbohydrates and fats, and that way you get started faster, but the fats will carry you out. So, energy storage is a function of fat. You can put there that we store fat in adipose cells. We store fat in adipose cells. The second major function of fat, I don't know why on your little sheet I have a blank and then 2, 3. I would just go on and put 1, change it, or put the first one as 3 or whatever. I don't know why I did that. The second purpose for fat is that it cushions the vital organs, cushions your vital organs, such as your kidneys. Now, I thought this was important, but now I know how important this is. Y'all have got to listen to this. 2 years ago, one of my students in class here told me that her boyfriend's mother was working at a 7-Eleven and that she got robbed. And the person who came in to rob her had a knife, and he stabbed her with a knife. And it was a very serious stabbing. And when she got to the emergency room, they told her that the only reason she survived was because she had enough fat to protect her vital organs. If she didn't have that fat, he would have gotten her vital organs and she probably would have died. Well, so when I heard that, my little mind started going, and I started thinking, you know, I probably should go eat something and make sure that my vital organs are protected. So, you know, when you look at me, please don't say, you know, she needs to lose a few weight. No, you look at me and you say, she is keeping herself safe. It's kind of like a little armor. So you need to think about that. Now, some of you I'm worried about. You need to stay away with people that have knives because I know they would get your, you know, your organ. So you might want to think about that. I promise you they told me that. So I take it seriously. And because I care about you all, I want to protect you. That's why I brought you those cupcakes that day. Okay. The third thing is that it insulates the body. Fats insulate the body. So those, that would be a good multiple choice question. So I'll give you another reason for fat. And Of course, these are the three that would, if I gave you the scenario that somebody gets stabbed and they survived, you think it was because maybe they could have had it. You know, I'm not gonna give you that scenario, but you won't forget that. You'll remember that, I know. Bless you. Okay, a type of fats or lipids that's very important to us are called phospholipids. Put a little star by number 1. That's the one I want you to know from here. Phospholipids. What do you think would be attached to this lipid group?
Cell membrane.
What functional? What, you're right, cell membrane. What functional group would be attached to a phospholipid? Yeah, the phosphate. So it's a lipid with a phosphate group, and the reason they're so important is because that is what our cell membranes are made up of. Phospholipids make up your cell membrane. Phospholipids, you have 2 fatty acids attached to your glycerol, and the third is attached to the phosphate group. Now, when you look at this, you can see this is one phospholipid right here. Here is the head, and here is the tail. Okay? The head right there is hydrophilic, means it loves water, but the tails are hydrophobic. You know why? Because they've got they're hydrocarbons. Hydrocarbons are nonpolar. They hate water. So what they're going to do is you've got this molecule that part of it is hydrophilic, part of it is hydrophobic. It's going to just like the part that doesn't like water, it turns its back, trying to get away from the water. So what it does, if this is the outside of the cell, there's a lot of water on the outside of the cell. There's a lot of water on the inside of the cell. So what these molecules do is they turn their tails, which are hydrophobic, inside, trying to get away from the water, and the part that likes the water orients to the outside. This is a picture right here, a drawing of your plasma membrane, of your cell membrane. And you know what holds it together? Not covalent bonds. The only thing that keeps that together is those hydrophilic interactions. Yes?
Can you please go back a couple slides? just-
Sure, be happy to. Tell me where. There or another one?
What?
Another one?
That's it.
Okay. We'll talk more about that in chapter 7. What you need to know right now is that phospholipids make up your cell membranes. Okay, that's what you need to know for that. Make sure you know that.
Okay, you're good.
There's another type of lipids that I want you to know. I want you to know steroids are lipids also. And just know that many hormones, as well as cholesterol, are steroids. So if I give you an example of a hormone such as testosterone, something like that, you should know that that's in the that would be classified as a lipid because it's a steroid. All steroids are lipids. Lipids characterized by the carbon skeleton consisting of 4 fused rings is cholesterol. Cholesterol is a precursor from which other steroids are synthesized. You know, we hear so much about too much cholesterol and how it causes lipid deposits in your blood cells that we think cholesterol is bad. But cholesterol really is something that we need, and we'll talk about more why it's needed in the cell membrane in chapter 7. But your liver will produce cholesterol because we need it. But if you eat, your liver usually produces as much as you need. But what happens is if you eat too much in your diet, that's when you start having the problem. That could contribute to cardiovascular disease again. Okay, anything else about lipids? Let's go to proteins. Proteins, are they considered macromolecules?
Yes.
Yes, the only ones that aren't considered macromolecules are your lipids. Proteins account for more than 50% of the dry mass of most, so proteins are huge. There's a lot of weight-wise in each cell. There are lots of different things that proteins do. Let me show you this chart that's in your book. First of all, know that all enzymes are proteins. Enzymes are chemical agents that speed up a reaction. We'll talk about enzymes more in detail, but all enzymes are proteins. There are some proteins that their purpose is support. Examples of this is like the proteins in your hair, other connective tissue. Some proteins are for storage. Some proteins are involved in transport, such as hemoglobin, because it's transporting oxygen. Some proteins are involved in the hormone proteins, such as insulin. That's a hormone secreted by the pancreas. Insulin's a protein. Proteins are receptors sometimes. Proteins have to do with contracting your muscles, such as actin and myosin. The last one says defensive proteins. Do you know that all your antibodies that your body makes are chemically proteins? So proteins have a lot of different jobs to do. You don't have to memorize this chart, but you need to be familiar with the jobs of proteins in case I give you a multiple choice like that, and I throw in something like a protein codes for the genetic information. You would know that's false, because proteins do not code for genetic information. Or, let's see, what else could I say? A protein is a major source of storage of sugar. You know, that's not true, because that would be your carbohydrate. So I've already told you about enzymes, and we've talked about enzymes just a little bit. Here's a slide that will show you, if you have sucrose, the substrate is the word for whatever the enzyme is going to work on. But here's your enzyme. If you notice, it's very specific. It's got this shape, and the only molecule that will bind to it, that will fit, is sucrose. This enzyme would be called sucrase. Enzymes always end in ASE. So this will bind right here, and we'll talk about later about how it really speeds it up. This will not make a reaction happen that's not going to happen anyway, but it will speed it up. Okay, we have water coming in, because if we're going to break down sucrose, we have to add water, right? Because this would be what kind of reaction? Hydrolysis? Are dehydration and hydrolysis essentially the opposite of each other? Yes, okay. So we're adding water. When that happens, you have the bonds that break between your glucose and your fructose. And now that this happens, then it's like these two individual molecules are released, glucose and fructose. Your enzyme is back just like it was, good as new, ready to go find another sucrose. So that's just a 5-second little talk about enzymes. We'll talk about more about this later. Okay, so if you have monosaccharides going together to make disaccharides, which will go together to make saccharides, polysaccharides, which are going to make carbohydrates, what is the building block for proteins? What goes together to make up the long chain? Yes, very good. Amino acids are the building blocks for proteins. Do you know how many different amino acids there are? 20. Now, if you have a polymer of amino acid, we call that a polypeptide. Poly meaning many. The reason they call it a polypeptide is because, what do you think the bond is going to be called? Skip down to number 7. The bond between amino acid groups?
Peptide bond.
Peptide bond, right. Now, a protein is several polypeptide chains hooked together, formed together in some structure. A protein is, I said that, it's a 3D structure. Amino acids, I said that they all have two functional groups? All amino acids have two functional groups, and they're all going to have an amine group, which is going to be this, and let's put a carbon there, and they're also going to have a C double bond O. They're going to have a carboxyl, and they're going to have an amino group. That's going to be on the test, and everybody's going to get that one. Nobody's going to miss it, okay, because I've said that enough. How they differ is whatever their R group is up here, that's going to make the difference in the 20 different amino acids. This is showing the skeletal structure of amino acids here. Some amino acids are polar, some are non-polar. These are the non-polars. This is showing you, sometimes they say GLY or just G. Most of the time, when I was in school, we always did the 3-letter abbreviation for these amino acids. You don't have to know these, but if you take cell biology or either biochemistry, either one, you'll have to memorize each of these structures of these 20 different amino acids, which ones are nonpolar and which ones are polar. I'm just showing them to you. And some are acidic and some are basic. So he said that they are connected with peptide bonds. If I tell you a structure has peptide bonds in it, and I said, what would be that compound? What would that be chemically? You would tell me it's a protein if it's got peptide bonds in it. A functional protein is not just a polypeptide chain. It's one or more polypeptides twisted and folded together in a certain way. Your book said that it's like, it asks this question, are threads of yarn, Y-A-R-N, synonymous with a sweater? Well, of course not. You can have just yarn by itself, and that's not the same as putting a sweater on. But if you take that yarn and you twist it and you fold it in a certain way, you can make a sweater. That's the same thing. You have, with amino acids, with proteins, what you do is you start out with amino acids, which you could have, here's one amino acid connected to another one, connected to another one, it could be the same one again, whatever, etc. These would be amino acids. They go together to make a polypeptide chain. Then it would be several of those polypeptide chains that would go together to make your protein. Now, each polypeptide has a unique sequence of these amino acids. There are 20 amino acids, but there are lots and lots and lots of different proteins. And the way you get a different protein is based on what these amino acids are. And depending on what these amino acids are, will determine the shape, and the shape will determine the function of your protein. This is showing you here. They've already made, before this, they showed this slide. Here's one peptide, amino acid, formed by a peptide bond. Okay, we've got these two amino acids. We have a growing polypeptide chains. We want to attach this amino acid. I know it's amino acid because I see an NH2, an amino group here, and I see a carboxyl group. So I want to attach this to this growing polypeptide chain. Anytime I'm building up, I'm going to split water out, which is what I did, which is the dehydration reaction. There'll be several questions on the difference in dehydration and hydrolysis. Now that I've done that, what kind of bond is formed? It's a covalent bond, but it's not going to be glycosidic. It's not going to be an ester bond. It's going to be a what? A peptide bond, yes. Then if we want to add another one up here, you know what we do? We split out water, the OH group here and an H from there, and keep repeating. That's what happens when you have the growing peptide thing. A specific protein structure determines how it functions. And I'm going to give you a real good example of that in just a minute. Okay, so if I ask you, is a polypeptide synonymous with a protein, you'll say no. This is showing you here, I told you that antibodies are all proteins. If you have an antigen, which like, let's just say it's a virus or something, and this is its shape, then the antibody that will render this ineffective will be exactly shaped, just like that. And this is the only virus that it's going to work on. If the virus is not shaped like this, it's not going to work, because it's got to have this to render it ineffective. So the shape of this is going to determine its function. That's what they're showing you here. This is a 3D picture of an actual antibody, and you see how this antibody is just going to fit like a little glove right onto that protein from that flu virus. Okay, there are 4 levels of protein structure. The first is called the primary. The primary is just what amino acids there are. Amino acid, amino acid. Then when you have secondary, you have 2 polypeptide chains, and you have some, this is a primary like this. You just have all these different amino acid subunits. Okay? Then when you have secondary, what happens is you have your amino acid subunits, but then you have some interaction between the chains. Then tertiary, we have interactions between the R group. So we have even more interactions in a different way, which is going to give us a 3D shape. Then we go to the next one, quaternary, which is going to be our final shape for our protein. Now, sometimes, this is showing you the hemoglobin, how that it can carry, that's the shape of it. Hemoglobin, you don't have to know that it's got two alpha and two beta chains. Okay, a slight change in primary structure can function a protein structure. In your space down at the bottom, jot this down. Sickle cell disease or sickle cell anemia. This is an example of if you change one amino acid, you've messed up the whole shebang. One amino acid in a chain of probably 1,000 amino acids is going to really mess this up. It's an inherited blood disorder, and it results from a single amino acid substitution in the protein hemoglobin. Now, if you look at this, we have the normal hemoglobin. They're just showing you these 7 right here because that's where the change occurs. Everything else is the same, they just didn't copy it. But if you notice, this is like amino acid valine, goes like that, threonine, proline. Look right here. There's a change. We have a substitution. One amino acid substitution. You know what that does? Because we have that substitution, that's got different R groups and everything. When we get the secondary and tertiary, it's going to have a different shape. So instead of being this shape, it's going to be this shape, which, because it's that shape, the problem is there's going to have other interactions in its quaternary structure and its final quaternary structure. This one, the hemoglobin, the way it's made, the molecules do not associate with each other. You've got this different kind of association where you've got plenty of space for the oxygen to be carried. This is a normal red blood cell full of individual hemoglobin molecules, each one carrying oxygen. But in this case, the way that the shape this is, they start attaching to each other. So the hemoglobin molecules clump together, and then that makes the red blood cells this shape, and they're deformed. They cannot carry as much oxygen because they don't have as much surface space. And not only that, that they stick to each other in your blood, which you know that's going to cause another problem. So if you inherit one of those alleles from one of your parents that's coding for the number 6 amino acid to be valine, then you will have sickle cell. If you just have one of the alleles, you know you have 2 alleles, one from each parent. If you just get it from one parent, then we say that you have the sickle cell trait. If you get it from both parents, you have sickle cell disease, which would be like this. If we say that this is normal, and this is sickle cell, so if you have, this would be how a person that's normal for hemoglobin, normal hemoglobin. If you got that trait from one parent, then you would have the sickle cell trait. And actually, in this case, half of your blood cells will look normal and half will be, like I can't really draw, let's say that's normal, then half of them will be like this, folded like that. So do you think a person who has blood like that is going to do well in a place like Colorado? You know, whenever you have less oxygen and you're going to need, or exercising, when your oxygen, anytime your oxygen is increased, your oxygen need is increased, they're going to have trouble. My students taught me this, there is a person named Ryan Clark who plays for the Pittsburgh Steelers. Have y'all heard of him? He has sickle cell trait, and when his team, the Pittsburgh Steelers, go to play in Denver, he has to sit out because he can't produce, he can't breathe enough. You can't really play football with oxygen on. You know, most of the time these people are okay unless they're exercising heavily, or if they were to go on a high mountain hike, they might have to take oxygen with them or something like that. But seriously, they said that he doesn't play in that case. Now, if you got it from both parents, then we would say you have the disease, sickle cell disease. And that means all your blood cells look like this. They're all sickle, and you can imagine the problem with that. as far as not having enough oxygen and the clumping that occurs. So that's very serious. What is all this cost from? One single cell amino acid substitution. Yes.
I don't get something, like earlier, did you say there's only one type of amino acid, or?
20.
There's 20, okay,
There's only one change in here.
there's one.
Yeah. 20, and the 20 different amino acids
So
are.
if you get one wrong, it screws it up.
Now, in this one, there's more than 20 amino acids. I don't remember how many amino acids there are in your chain. But in this, sometimes you can have a substitution. This is caused by genetic. This is what it's coding for. Somewhere along, way back, I guess there was a mutation or something. But there was a mistake in the copying somewhere. I don't remember how many amino acids actually are in hemoglobin, but we're starting with 20 different subunits, you know what I mean? And sometimes there could be a mistake and you have one, and it may not cause a major problem, but in this case, you get the one amino acid wrong, which is probably each amino acid. Remember how we talked about in DNA, the bases, which we'll talk about in a minute, where you have like A, T, T, G, G, whatever, C, whatever it is. When you do this, you read these as a triplet code. It takes 3 of these nucleotide bases. That codes for some amino acid. I don't know what it is. Let's say that codes for methionine or something. And every time that the ribosome reads ATT, it's going to put that amino acid. Then it comes to GCA or whatever that is. And maybe that's glycine, I don't know. But that's how it attacks. So, you know, actually, you could have a point substitution here, where if that got changed to T, then you see that's going to be a different amino acid it's going to code for.